Greetings and welcome to the second lecture in our series on microbiology introductions. On the last session we talked about the essential structure and anatomy of bacteria, especially as it relates to the structure of the cell wall and the cell membrane. And today I wanted to go more into the identification and classification of bacteria. So now the way we're going to approach this today is much in the same way in which microbiology lab will go through in their process of identifying an organism that's growing on one of their plates. We're going to cover the gram staining technique, morphology, and then the variety of different chemical reactions that are used in both the identification and classification of bacteria. Now, in a previous seminar, I alluded to the gram stain as especially as it's relevant to the cell wall. And today I want to give a little bit more detail about gram stain technique so that you understand both how it works and why the cell wall structure is such an important component of the identification and classification of bacteria. The gram stain is the most important staining procedure used in medical microbiology. There are a variety of other stains that are used to help identify organisms, but the vast majority of bacteria are classified using the gram stain, and so it's usually the first step after a specimen has been received. The microbiologist will take the specimen and will usually take a sample and apply it to a slide using a sterile loop, and then fixate it to the slide, sometimes using heat or just using direct microscopy depending on the specimen. And then we'll apply a series of stains to differentiate gram-positive from gram-negative bacteria that are present in the specimen. The first step in the staining is to use a crystal violet stain that is washed over this specimen, and then an iodine solution is used to fixate the crystal violet or to crystallize it inside the cells. Now, the next step is then the use of an alcohol wash that, depending on the cell wall structure, will either wash out the crystal violet inside the cells, or the crystal violet will be retained. The final step is then to use a pink dye to stain all of the cells that were not counterstained with the purple-violet. So on a specimen, such as you would see in this example, you can see two different groups of organisms that have been stained. Those that are stained purple and those that are stained pink. Now, the gram-positive organisms retain the crystal violet stain and so will stain purple, and that's why we call them gram-positive. Whereas the crystal violet is washed out of gram-negative bacteria, and so it is counterstained using the pink, and so a gram-negative organism will stain pink on a slide. Now, as I mentioned, there are a number of other staining techniques that may be used depending on the organism, but for the purposes of these sessions, we're not going to explore those. An example of a special stain is the acid-fast staining, which is used to identify a mycobacterium, which has a different cell wall structure, and so doesn't actually respond to any of the stains used in the gram stain. So now that a specimen has been stained, a microbiology tech can now look at the specimen under a microscope and actually start to see all of the different morphologies that may be present to help it identify the bacteria. Now, this slide shows you a series of the structures that can be seen and we largely break them down into either those bacteria that appear circular or spheroid in shape, called cocci, or are more elongated and rod-like, which we then refer to as bacilli. There are a variety of other morphologies that may be seen depending on the species, but again, for the purposes of this series, we're not going to talk about those more unusual bacteria and focus more on the spheroid and the rod-shaped bacteria only. Now, it's not only the shape of the bacteria that helps in its identification, but also in the way it aggregates that gives a clue to its final identification. So, for example, with coccus bacteria, they can generally either be single, as single coccus, or they can be diplococcus with where they pair up, or they can form into clumps, and that's very typical of Staphylococcus, hence its name. And so the grape-like appearance of them is a clue to the genus of that bacterium right out of the shoot. Or it can appear in a long strip, or Streptococcus, and again, this is the genus name for that group of bacteria. And so, seeing a strip of spheroid bacteria gives you a pretty good sense that this is probably going to be a Streptococcus. The presence of the staining can also help identify encapsulation, which is a clue to the presence of a pneumococcus or Streptococcus pneumoniae, which typically appears as pairs of spheroid bacteria with a mucoid or a capsule covering them that they will show up on the stain as clear. Within the Bacillus group, you can see bacteria form a variety of different aggregations. They can be shaped as in a simple rod like a bacillus, or they can be coccobacillus, as can be seen with Neisseria, or they can form into diplobacteria or streptobacilli, which are not as common, and the vast majority of bacillus bacteria usually are occurring as single individual organisms without any sort of association or aggregation. But right from the very beginning, once you have a stain and you can see not only the morphology of the organism, you can also see how they're aggregating or organizing themselves to give you additional clues on the identification. Now, at this point, the microbiology technician will inoculate the specimen onto a agar plate or into a culture medium to utilize its growth characteristics and its appearance to help further identify and specify which organism they're dealing with. And this is the point in which a series of chemical reactions can be used to help with the identification of the organism. The chemical reactions that we're referring to are not chemical reactions that we inflict upon the bacteria, but are actually the chemical processes that are inherent within the bacterium that differentiate different species from each other. Because not all bacteria are capable of growing in the same environments, or may lack certain metabolic pathways that others may have, and we take advantage of these differences to help in the identification. So the first chemical reaction that's important to help us in the identification is whether or not the organism can thrive in an aerobic or oxygen-rich environment, or whether it requires an anaerobic environment. There are certain species of bacteria, particularly the Clostridium family, which are strict anaerobes and have to live in an anaerobic environment with absolutely no oxygen. And so one way we can help identify whether the organism is aerobic or anaerobic is to differentially plate, meaning putting a sample on a plate that's in an aerobic environment and then another sample that's placed in an anaerobic environment. Blood cultures take advantage use this method, and this is why the blood culture is collected in two vials. One of it is an anaerobic, and the other is an aerobic environment, and this will help more rapidly identify an organism, because if it's an anaerobic organism, it will only grow in the one bottle, but if it's an anaerobic, or aerobic rather, it will only grow in the other one. So this helps with the identification. Now alternatively, you can use a method where you just take a specimen and plate it across a tube of agar, as you can see here, and then just actually look to see where the organism will grow. So in the first tube, you'll see all the organisms tend to cluster near the top of the agar, and this tells you that this is an organism that requires an oxygen environment in order to survive. Whereas in the second tube, the bacteria all seem to be clustering at the bottom as far away as possible from oxygen, suggesting that they're anaerobic bacteria. Both of these are considered obligate anaerobes and aerobes, meaning that they absolutely cannot grow in either an oxygen-rich or an oxygen-poor environment. They need the presence or absence of oxygen in order to survive. There are other organisms which are facilitative, which means that they can grow with or without oxygen. And so in group 3, or tube 3, you can see this would be an example of a facilitative anaerobe, meaning that it can grow in any environment. It can grow in an anaerobic environment, as you can see. Some of it is growing at the bottom of the tube, but it tends to cluster near the top, meaning that it actually wouldn't mind being in a more oxygen-rich environment. And this is because aerobic respiration, as you recall, generates more ATP, and so it gives you much more of an energy advantage to be an aerobic organism. But if push comes to shove, it can live in a anaerobic environment, but then relies more on fermentation and glycolysis in order to survive. Tube 4 is a group of organisms that are called microaerophiles, which means that they use oxygen but are poisoned by high concentrations of oxygen, as you would see in in the normal atmosphere. It's too much oxygen, and they're actually poisoned by that higher concentration, and so will tend to settle in a lower down in the agar, where the oxygen concentrations are a little bit lower, and that it's much more able to survive in that. The final tube is what we call the aerotolerant organisms. Now, these are organisms that do not require oxygen at all. They don't use it to metabolize energy, and they rely completely on anaerobic metabolism in order to survive. Now, these are different than the obligate anaerobes that you saw in tube 2, because these organisms would not be poisoned by the oxygen. Obligate anaerobes are not only do they not like living in an oxygen survivor, they actually are killed by the oxygen. It's too toxic for them. Oxygen by itself is a very toxic molecule that is responsible for the vast majority of the weathering and destruction you'll see in the environment. Rust, corrosion, these things are all a byproduct of oxygen. We have all developed as eukaryotes, we've all developed systems to manage it, and in fact take advantage of the reactivity of oxygen in order to up our ability to metabolize and generate energy. But for bacteria, they don't have as many of those metabolic engines, can't take advantage of oxygen as much as eukaryotes can, and then can sometimes be consider oxygen the toxic the molecule that it actually is. The most important, medically important organisms are usually either strict anaerobes or strict aerobes, and so the differentiation between facilitative anaerobes and obligate anaerobes and microaerophilics is not as important. It's more for your own interest. So now that we have an gram stain as well as a structure, we can start differentiating them. Starting with the gram-positives, we can break the gram-positives into those that are gram-positive cocci and those that are gram-positive bacilli. The gram-positive cocci form the majority of the medically important bacteria, and so I'm going to focus a little bit more of those next. The gram-positive bacilli are the groups that include Clostridium, most concerning, so be aware if you get a gram stain that says gram-positive bacilli in a patient that's septic, this is probably a Clostridium species, most concerningly Clostridium perfringens, and you need to act quickly. But there are other gram-positive bacilli, both Corneobacterium, which is more of a skin organism, Listeria, and Bacillus, which are all have the same staining and morphology. The vast majority of gram-positives that you're going to encounter, though, are coccus-shaped, and so the differentiation there is whether or not it's a Staphylococcus or a Streptococcus. Now, initially on its how the organism is arranged on the slide, whether it's in strips or in clumps, gives the microbiology tech a leg up clue as to what organism they're dealing with. But that's not always necessarily the most sensitive method of identifying. You will often get a gram stain report, which will read to the effect gram-positive cocci in clumps or gram-positive cocci in chains. And this gives you the clue that this is either a strep or a staph species that you're going to be dealing with. At the same time, though, they're going to do further identification of the species so that they know exactly what they're dealing with. And so the first step in this is to take a sample and expose it to peroxide and see whether or not the organism has catalase, an enzyme that can break down hydrogen peroxide present in it. If the organism has catalase, then this makes it a Staphylococcus. The next step after that chemical reaction is to test to see whether the organism has the presence of coagulase. Because if it's coagulase positive, then that makes it a Staph aureus. And then if it's coagulase negative, which again you will frequently encounter coag-negative staph, which is a much less virulent organism, in general forms the staph epidermidis and saprophylicus groups, of which I'm not going to get any further differentiation on. But more importantly, the Staphylococcus that is coagulase positive is Staph aureus, and in order to identify that organism, you need to go through two different chemical reactions to identify it. Now, the Streptococcus is much more complicated than that. Now, this is the catalase negative group, but the next chemical reaction is how it reacts in the presence of blood. So the organism has to be plated on a blood agar plate, and then how it metabolizes blood helps in the differentiation of this individual species. If the colonies that form on the plate consume the blood that's present in the agar, then they will form a rim of clear agar around it. So it uses the blood. We call this the beta hemolytic organism. The most important organism in this group is the strep pyogenes, or group A strep, which is confusingly also a beta hemolytic organism. If the Streptococcus doesn't react to the blood at all, doesn't do anything with it or consume it, then this is called a gamma hemolytic organism, and this is the Enterococcus group's of which are the Faecium and Faecalis are the two species of note. But then if the organism only partially metabolizes blood and then leaves essentially a green ring around each of the colonies, these are called the alpha hemolytic group. And this is the pneumococcus or streptococcus pneumoniae and the viridin groups, of which you can then do additional testing on bile solubility, the presence or absence of a capsule, capsules present on pneumococcus, and also sensitivity to certain antimicrobials as clues as to its identification. Now let's say you have a gram-negative organism on your gram stain, and you look at it under the microscope and you see that it can either be a coccobacillus, a cocci, or a bacillus shaped organism. If it's coccobacillus, then you're pretty certain that you're now dealing with a Haemophilus species, a genus, and then it's just a question of that species. But in general, Haemophilus, very commonly found in the respiratory tract, easy to identify, and further identification beyond this is not always necessarily pursued. The next step, once an organism has been identified, is to then check its sensitivities to the variety of antibiotics available, and this is a completely different step done as the final portion of the investigation in the microbiology lab. The identification of resistance and the mechanisms of resistance are not covered in this series and are covered as part of the infectious disease section that others will be putting on over the course of the next years. Another concerning gram-negative that you may occasionally hear about and get an initial gram stain report on is a gram-negative cocci. And a gram-negative cocci should immediately raise suspicion for Neisseria genus, and Neisseria meningitis is the species of biggest concern. This is a cause of meningococemia and meningitis, and so in anybody who you get a phone call from Microlab saying initial gram stain shows gram-negative cocci, you have to worry about meningitis in that group. Neisseria also has another species, gonorrhea, which is obviously an important organism in sexually transmitted diseases, but less so in a critical care environment. We're not often encountering patients that are floridly septic from gonococcus. The vast majority of gram-negatives are bacilli in shape. And so this is why we often just group them all as gram-negative bacilli, or GNB as sort of the abbrevium that we use for this group of bacteria. Now, these organisms are generally aerobic bacteria, so the oxygen sensitivity doesn't help in the identification any further. And so then we take advantage of the metabolic pathways that are available within the variety of bacteria within this group to help better identify it. The first differentiation that is used is whether or not the gram-negative bacilli is a lactose fermenter or a lactose non-fermenter. If it can not use lactose as an energy substrate, then the next step is to check whether or not it reacts to oxidase. Because if this is positive, then this is Pseudomonas, and if it's negative, then it's probably another enteric like Salmonella or Shigella. If the organism is a lactose fermenter, and again on an initial report after the gram stain, the next day you'll probably get an update that the gram-negative bacilli is a lactose fermenting gram-negative bacilli. The next step is to sort out whether this is a Klebsiella, E. coli, or Enterobacter, or whether it's a Citrobacter or a Serratia. And the way the distinction is made is by utilizing whether the speed with which the lactose was fermented. Slow fermenters tend to be the Citrobacter and Serratia, whereas fast fermenters are the Klebsiella, E. coli, and Enterobacter. In order to further identify to identify the organism, you then do two other metabolic tests. If the organism is urease positive, it produces urease, then it's a Klebsiella. If it's indolin positive, then it's an Escherichia or E. coli, and if it's neither of those, then it's Enterobacter. Now, at the end of the day, this identification process can take, well, many days to be accomplished. It requires that the organism is plated on a media that it can grow on. It needs to then go through a series of chemical reactions, and the chemical reactions take time. And then after that, we have an identification. It's still going to take an additional few days to test its sensitivities. And so having a full culture and sensitivity of any sample can take upwards of 3 to 5 days before you get results. And obviously that's the reason why a lot of empiric therapy is used in antibiotics. But fortunately, on the horizon are a number of different techniques that utilize DNA probes and even mass spectroscopy to more rapidly identify organisms, to better tailor therapy, and to provide vital information as soon as possible. So while a lot of these tests still exist in a lot of most microbiology labs, over the course of the next decade, expect to see faster identification and more accurate identification as the use of genetic and other techniques come into the forefront as methods of identifying these organisms. So that's basically an overview of the identification and classification of medically important bacteria. This, in combination with the first lecture on the bacterial structure and anatomy, gives you a general overview of basic microbiology. In the next series, I'm going to delve into more detail on the medically important bacteria as groups, the Streptococcus, the Staphylococcus, and talk in more detail about these individual organisms, not only their characteristics, but also the types of infections they cause, to make this whole thing a lot more clinically relevant for you.